Alright class, let's continue with the cell envelope. The cell envelope is one of those hard to define areas. Is it part of the cell? Is it in the cell? Is it out of the cell? It's that nebulous area that separates the inside from the outside. It's outside of the cytoplasm, but it's still technically considered part of the cell. So if we look at our cell envelopes, we're going to have always 2 or sometimes 3 essential layers to them. Every bacteria is going to have a cytoplasmic membrane or an inner membrane. Every bacterium is going to have a cell wall, but some bacteria, particularly gram-negative bacteria, are going to have an outer membrane. If there is no outer membrane, generally speaking, it's going to be considered a gram-positive bacterium. So here we go. Here we have a gram-positive bacterium with no outer membrane. It has the inner cell membrane or plasma membrane, and then it has a nice thick cell wall. And this entire package here, the cell wall plus the cell membrane, is considered the cellular envelope. And the same is true over here with this gram-negative bacterium. It has an inner membrane, and then it has a thin cell wall, and then it has an outer membrane. One thing that's notable about the outer membrane of a gram-negative bacterium is that it has lots of excuse me, late night. It has lots of lipoproteins and lipopolysaccharides embedded in its outer membrane. The cell wall of the bacterium is primarily responsible for determining the shape of the bacterium. The cell wall of the bacterium can be reformed based on the metabolic processes of the bacterium. The cell wall of the bacterium also gives it a lot of structural support to keep it from lysing under osmotic pressure or from collapsing under hydrostatic pressures. The drugs that target cell wall reproduction and reproduce and disrupt the ability of the cell wall to be manufactured are going to cause lysis of the cell, or cause the bacterial cell to rupture by compromising the integrity of the cell wall. The cell wall of the bacteria is constantly accumulating small structural deficiencies, and the bacterium has to constantly repair the cell wall. It's kind of like a highway system or a road system. The moment you stop doing maintenance, that's when things start falling apart. And the same is with the bacterial cell wall. Some antibiotics prevent the bacterium from performing maintenance duties on their cell walls, and then the bacterial cell wall breaks down and the bacteria pops. Our bacterial cell walls are made up of those subunits of N-acetylglucosamine and N-acetylmurmuric acid, or the naganams. So the naganams are abbreviated with these M's and the G's, and these are special carbohydrates that are linked together with glycine interbridges. So where we see this chain right here, this yellow line, this is a peptide that's linking together the polysaccharides that make up the cell wall of the bacterium. This is a unique complex carbohydrate for bacterial cell walls. So when a patient takes an antibiotic that targets cell wall repair or cell wall synthesis, generally speaking, that antibiotic, that drug, is preferentially going to affect bacteria and not affect human or eukaryotic cells, because we don't have these peptidoglycan polysaccharides making up our cells. And we look at our gram-positive bacteriums. They have a very thick sheet of peptidoglycan to make up their cell wall. This thick sheet of peptidoglycan is generally between 20 and 80 nanometers in thickness. It can be a little bit more, a little bit less. This is an average range. It also contains 2 notable chemicals, teichoic acid and lipoteichoic acid. These 2 chemicals are going to function for cell wall maintenance and enlargement and make the cell wall have an acidic charge. Now when I say acidic charge, I mean that the cell wall is going to be very positive in nature. If we look at our gram-negative cell walls, these gram-negative cell walls are going to be much thinner. If we flip back a couple slides here, we can see that in the gram-positive cell wall it's very thick, and in the gram-negative cell wall it's very thin. The thinness of the gram-negative cell wall gives the gram-negative cell a lot more flexibility, and on the flip side, though, makes the gram-negative cell much more susceptible to bursting or lysing because it doesn't have a thick coating around it to protect it. During the Gram staining process, and this is a classic staining process, we start off with staining the cells with crystal violet, and then after we've stained our cells with crystal violet, we need to add iodine, Gram's iodine, as the mordant. After adding Gram's iodine as the mordant, causing the crystal violet to stick to the peptidoglycan, we rinse off the excess crystal violet with alcohol, and if we have a gram-positive bacterium, the crystal violet will stay stuck in the peptidoglycan layer during this stage, but because a gram-negative bacterium is surrounded by lipids on its surface, the crystal violet washes off with the alcohol during this stage. And then at the very end, we have our counter stain. We'll use safranin and stain the cells with safranin. The gram-positive cells will pick up that purple or the pink safranin, but they've also been stained dark purple, so they remain that dark purple color. And then the gram-negative cells will now be colored pink. So if you're looking at a photomicrograph and you see a lot of purple and pink cells, that's a good indication that it's a gram stain of a mixed culture. Sometimes we'll have something that falls outside of the range of gram-positive and gram-negative, gram-indeterminate or non-typical. These are going to be acid-fast bacterium. Good examples include Mycobacterium, the genus Mycobacterium, and the genus Nocardia. These bacterium have a lot of mycolic acid in their glycocalyx. Mycolic acid is a long-chain fatty acid that's found in the cell walls of acid-fast bacteria. It makes acid-fast bacterium highly pathogenic or highly virulent because it causes these bacterium to be difficult. It makes it difficult for these bacterium to be phagocytized. It also makes it difficult for us to stain the bacterium. If we cover something in wax, it's going to be difficult for us to have a liquid penetrate the wax and interfere with the cell that's been covered with that wax. Another example of a non-typical cell wall would be archaeal cell walls. These cell walls typically are not going to have the glycine cross bridges in them. Other times, though, we'll have cell walls that are made of pure protein. Depending on the species of Archaea, you have a lot of variety or variability among the cell walls. One thing that I want to emphasize for you, though, is that archaeal cell walls do not have peptidoglycan. Bacterial cell walls do have peptidoglycan. Archaeal cell walls do not have peptidoglycan. Some Archaea don't even have a cell wall. They just have a cell membrane. Sometimes, if we look at our bacteria that do not have cell walls, we will see what are known as Mycoplasmas and L forms. A Mycoplasma is something that naturally lacks a cell wall. Instead, there will be a lot of sterols in the cell membrane that stabilize the cell against lysis. These sterols are going to increase the viscosity of the cell membrane and make it stiffer and more resilient to impact so that it's capable of protecting that cell. A good example of a mycoplasma would be Mycoplasma pneumoniae, aka the pathogen that causes walking pneumonia. We also have L forms. These are very poorly understood. They're kind of the new kids on the block, so to speak. These are bacteria that naturally have a cell wall but can temporarily lose their cell wall during some part of their life cycle. So they can alternate back and forth between having a cell wall and not having a cell wall. Obviously, since a lot of our antibiotics are designed to target cell wall synthesis, if we have a bacterium that can switch back and forth between having a cell wall and not having a cell wall, that bacterial species is going to be very resistant to a wide variety of antibiotics. And consequently, these L-form bacterium play a role in persistent infections in human beings. If we look at gram-negative bacterium, they have that outer membrane outside of the cell wall. The membrane is going to be similar to the plasma membrane. You can think of it as an additional plasma membrane. There's going to be a few differences, though. There are going to be some lipopolysaccharides in the outer membrane. These lipopolysaccharides are going to be identification molecules. They signal what the cell is, and they also serve as receptors for picking up and conjugating the cells, or picking up extracellular chemicals. These lipopolysaccharides also can function as an endotoxin. An endotoxin is a toxin that is going to remain in the bacterium until the bacterium dies. One of the major concerns about fighting off a gram-negative infection is if you have a high concentration of gram-negative bacterium and you kill them off too quickly, those gram-negative bacterium will start to lyse and then release lots of lipopolysaccharides into our bloodstreams. Lipopolysaccharide is a toxin. It causes quite negative side effects. As long as the gram-negative bacterium remains intact, that lipopolysaccharide remains trapped in the gram-negative bacterium and is not released to us, the human beings. It's when we kill off lots of gram-negative bacterium very rapidly in our body that we worry about toxic shock from lipopolysaccharide. The outer membrane of gram-negative bacterium also have porin proteins. These porin proteins you can think of as a special hole that allows water to go in and out. Every once in a while, these porin proteins will all be designed to allow other chemicals to go in and out. The most common, though, is going to be an aquaporin, a porin for water. If we look at the cytoplasmic membrane, or that inner membrane that's in both gram-positive and gram-negative bacterium, this is going to be your classic phospholipid bilayer with embedded proteins. This is going to serve the site of energy reactions. So if you've taken physiology, you've learned about glycolysis, the citric acid cycle, and the electron transport chain. And here in the cell membrane of bacteria, we have almost identical energy processes or reactions to the electron transport chain. We also have nutrient processing and molecular synthesis. So we are going to make new molecules for the bacterial cell in the cell membrane. The cell membrane is the regulator. It regulates what goes in and out of the cell, including nutrients and waste. And because it's going to be picky about what it lets go in and out, it's known as selectively permeable. Some things can go in, some things can go out. It's choosy, it's selective, it discriminates, so to speak, against what it allows to go through. If we look at the cell envelope in general, and this is going to be the part of the bacterial cell that separates the inside from the outside environment, the intracellular environment from the extracellular environment. If we look at gram-negative bacteria, that they have that extra outer membrane compared to gram-positive bacterium. And because they have that extra outer membrane, will have one extra hydrophobic region that a drug will need to penetrate. And because of that extra lipid layer, it's very difficult for antibiotics to penetrate gram-negative bacterial cell envelopes. Because it's more difficult for those antibiotics to penetrate, it's much more difficult to fight off in a gram-negative infection with traditional antibiotics. However, if you are not fighting off the infection per se, but you're trying to sterilize the surface, alcohol is particularly effective in killing gram-negative bacterium because alcohol is an excellent dissolver of phospholipids. Alcohol groups have both polar and nonpolar regions, so if we use an alcohol swab to cleanse our skin, we will preferentially be destroying gram-negative bacterium just because they have those lipids on their surface. If we are going to use medications to treat a gram-negative bacterium, those medications need to be designed to penetrate not only the cell wall and the inner membrane, but they need to penetrate that outer membrane that's on the gram-negative bacterium. So this is going to be a very elite class of medications that can penetrate those hydrophobic and hydrophilic regions of the cell envelope of a gram-negative bacterium. So, concept check. If we look at the outer membrane of the outer membrane contributes an extra barrier in gram-positive bacteria to make them impervious to some antimicrobial chemicals. So they are generally more difficult to inhibit or kill than gram-negative bacteria. Well, hopefully you paid attention on the last two slides, but if you didn't, you can rewind or flip back. I'll pause. You should pause now and look up the answer. 1, 2, 3. The answer is false. Gram-negative is more difficult to kill. Gram-positive is much easier to kill. Hopefully you got that right. If you have any questions, shoot me an email or post it on the discussion board. Happy studies.